Acceptable intake limits for nitrosamine impurities in the drug products. This is the topic for the video. Nitrosamine impurities are the impurities which have very stringent limits. As these nitrosamine impurities are carcinogenic in nature, mutagenic in nature, so these are required to be controlled. The AI limits means the acceptable intake limits for nitrosamine impurities are stringent because of the nitrosamine impurities potency. Understanding of the AI limits is must to know for the pharmaceutical professionals and mainly who are working in the pharmaceutical industry. Nowadays, the regulatory authorities are very stringent and they are observing these impurities in the products very closely. Many of the products got recalled from the market because of presence of the nitrosamine impurities, and I hope you might be known to these terminologies and you might be known to the nitrosamine impurities. I have made many videos and these videos are present in the playlist, so I request you to go through those videos to have the complete understanding about this topic. This video is for the AI limits. So be with me till the end of this video and you will get good information for the AI limits. For making this video, I have referred FDA guidelines, EMA guidelines and ICH M7 guideline for making the AI limits information. Nitrosamines, as we have seen, these are the chemical impurities which may form during storage or which may arise from manufacturing or may come from the contamination. Some nitrosamines are highly mutagenic and carcinogenic, and that's why they can pose a potential cancer risk. Regulatory bodies like EMA, US FDA, and ICH provide the guidelines for the control and limiting the nitrosamine impurities in the APIs and the drug products. So nitrosamine impurities are classified as small nitrosamine impurities and the NDSRIs. That means two types of nitrosamine impurities are there which are required to be controlled. Control the intake of nitrosamine to reduce the risk of cancer, that is 1 in 1 lakh species for the lifetime risk threshold. Now you might be thinking or you might have a question like how these AI limits are derived. What is the science and justification behind defining or behind the AI limits? So there is a definition of AI limit. AI limit means acceptable intake limit. It is a level of daily exposure to a mutagenic impurity that corresponds to an increased cancer risk of one additional case in 1 lakh subjects over a 70-year lifetime. So by definition, if the product is given to the patient over a period of 70 years, considering the lifetime of 70 years, and if that product is contaminated with nitrosamine impurity, and if the lifetime exposure of this impurity to the patient has a risk of cancer to one subject in 1 lakh subject or 1 in 1 lakh subject, that means there is a risk and the impurity is required to be controlled at that level. That's why these impurities have very stringent limits. The AI limits are calculated based on exposure assumptions in both drug substances and the drug products. Now another terminology is TTC. TTC is nothing but it is a threshold of toxicological concern. For unstudied chemicals, an AI of 1.5 microgram per day is considered acceptable for negligible carcinogenic risk or negligible toxic risk. So always remember the TTC is 1.5 microgram per day. That means 1500 nanogram per day. Highly potent mutagens, these are called as the cohort of concern, and these highly potent mutagen includes the compounds like N-nitrosous compounds, which could present significant cancer risk even at low intakes. So it is not like all the compounds may have the limit of 1500 nanogram per day. Some potent mutagenic nitrosamine compounds are there which are required to be controlled at much lower levels. Now we will see what is the carcinogenic potency categorization. Till now you have understood or you have learned like what is AI limit, on what basis the AI limit is calculated, how it is calculated, what is the justification behind that. Then we saw the TTC base AI limit, that is 1500 nanogram per day. Now we'll go ahead and see the other approaches. So CPC approach is there, that is carcinogenic potency categorization. So when to use this carcinogenic potency categorization approach? So in the absence of robust data, if you don't have the carcinogenicity or mutagenicity data for specific nitrosamine, that time use the FDA recommended carcinogenic potency categorization approach. There is a CPC approach given by the regulatory authorities that is required to be used in the absence of carcinogenic data. And this CPC involves a risk prediction based on known data from similar compounds or using other scientifically justified approaches. Approach considerations are bacterial mutagenicity data, in vivo testing, and read across analysis. So read across analysis involves using the structurally similar compounds, and this approach can help to estimate the carcinogenic potential of an impurity. Now we'll see determining the AI limits for nitrosamines, recommended AI limits for the nitrosamines. FDA provides AI limit for known nitrosamine impurities on the nitrosamine guidance web page. NDSRIs, these are the nitrosamine drug substance related impurities, and these NDSRIs may lack data, making it harder to set the AI limits directly. As I have mentioned, there are two types of nitrosamine impurities, small nitrosamines, which are like NDMA, NMEA, NMPA. You might have heard about these 7 nitrosamines, and NDSRIs. So NDSRIs are the impurities which have structural similarity with the drug substance. That's why they are named as NDSRIs. Then in case of data deficiency, so manufacturers can apply the CPC approach, that is carcinogenic potency categorization approach, and consult FDA guidance, or they can contact the agency for approval. Manufacturers must justify proposed AI limits with scientific evidence if they differ from the FDA recommended values. So CPC approach is to be used, and the justification is required for the different limits. Managing multiple nitrosamine impurities. So it is not like that every time in every product you will have single nitrosamine or single NDSRI. Sometime you might have two NDSRIs, two nitrosamines, or one NDSRI, one nitrosamine, or two nitrosamine and two NDSRIs. So anything can happen. If the product is sensitive to the nitrosamine impurities, then there are chances that multiple nitrosamines may be present. So when multiple nitrosamines are present, total nitrosamine exposure should not exceed the AI limit for the most potent nitrosamine in the product. Once you know that there are multiple nitrosamines, then your focus should be to see which is the most potent nitrosamine in the product, and that limit should be applied. Then handling variation in the potency. So if recommended AI limits for different nitrosamine vary significantly, an alternative flexible approach can be used. Total exposure has another approach which is given in the guideline. Use the approach described in Appendix C of the FDA guidance to ensure total nitrosamine exposure remains within the acceptable cancer risk limit. So, whenever you are facing the challenge of multiple nitrosamines, then always remember that the total risk should not exceed the risk of 1 in 1 lakh species. So, if you have 2 or 3 nitrosamine impurities, divide the risk and the total risk should not become 100%. I have made a separate video on the total nitrosamine impurities limits. You can go through those videos and you will understand how the total impurity limit is given for the nitrosamines. There are two approaches, fixed approach and the flexible approach. So once you go through those videos, you will clearly understand the scientific justification and the regulatory requirements. Now we'll see analytical testing for nitrosamine impurities. So as the nitrosamine impurities are very potent and they have stringent limits, so the analytical techniques are required to be very sensitive. Analytical method must detect nitrosamine impurities at or below the AI limit, and AI limit is given in nanogram per day. Limit of quantitation LOQ. LOQ should set to match the required AI limit. They must be scientifically justified to ensure accurate measurement. So, what is the limit of the nitrosamine? Based on that, LOQ is required to be set. FDA validation guidance. The ICH Q2R1 validation of analytical procedures provide guidelines for developing and validating methods for detecting the nitrosamines. The examples of validated methods, FDA nitrosamine guidance web page include validated methods for nitrosamine detection. AI limits conversion and units. So this is the example for calculation of the AI limits in nanogram per day. And AI limits are usually provided in nanogram per day. TTC based approach involves 1.5 microgram per day. You can convert it into nanogram and it will become 1500 nanogram per day. So how to convert the ppm from ng per day? Conversion to parts per million, that is ppm, to control purposes or for giving the specification limits, AI limits can be converted to ppm based on the maximum daily dose. So always understand that if your maximum daily dose is higher, that time you will have the very stringent limits, and if the maximum daily dose is higher lower, then you can get some relaxation or higher limit. Formula is given, ppm is equal to AI divided by mdd. So AI is in ng per day and mdd will be mg per day. If the AI limit is 26.5 nanogram per day, MDD is 50 mg, the ppm concentration will be 0.00053 ppm. So this is the calculation. Now coming to the default AI limits when no data is available. So what you will do if you don't have any data? Then which limit is required to be taken further? Default AI limit in cases where AI limit cannot be determined using the described approaches. Take a limit of 26.5 nanogram per day, which is a default AI limit. And as per my understanding and knowledge, the default AI limit may be different for different regulatory bodies. So whenever you are working onto the product, always check what is the default AI limit by specific regulatory authorities. Also check the MDD so that you can understand the calculation. Also refer whether the formulation is being given to the patient for a short period of time or for a longer period of time. That means whether the molecule is being used for acute treatment or whether the molecule is being used for the chronic treatment. Consulting the FDA. So manufacturer can request clarification or approval from the FDA if AI limit is not provided for the specific nitrosamine. And not only the FDA, other regulatory authorities can also be contacted and they can guide you further. So conclusion and regulatory strategy. What are the manufacturer's responsibility? And as required, the to manufacturers are required to ensure that acceptable limits of nitrosamine impurities are adhered and the proper care is taken to reduce the nitrosamine presence. Implement scientifically justified control strategy for managing the nitrosamine exposure. Regularly consult FDA guidelines and other country-specific regulatory body guidelines, especially for the new or unknown nitrosamine impurities. Some groups are there for scientific discussion and to resolve the queries. Then nitrosamine impurity related database is also there that can be studied and that can be referred. Now coming to the flexible approach. So use the carcinogenic potency categorization approach, that is a CPCA approach, and alternative methods when necessary. Continuously validate analytical methods to ensure accurate testing and control. So this is the information regarding AI limits for nitrosamines. Always understand your product, its maximum daily dose. Then do the literature search to know whether the molecule is sensitive for the nitrosamine impurities, whether the formulation is sensitive to nitrosamine impurities, is there any chance that the nitrosamine impurity may get formed, and based on that try to do the risk assessment, and based on the risk assessment try to decrease the chances of nitrosamine impurity presence, because these nitrosamine impurities are the potent carcinogen, potent mutagen, and the presence of nitrosamine may lead to incidence of tumor. So to protect the public health, the manufacturers are required to strictly adhere to the nitrosamine limits. I hope this video will help you to understand about the AI limits. If you have any queries, please comment and I will try to answer. Thank you for watching the video.